Chapter 11. So this chapter is all on our muscular system, but for this chapter, it focuses mostly on our skeletal muscle system because you have more ability for us to study that system, partially because it is more external and we're able to easily stimulate it. So this first section will go over just generic stuff about muscles and what defines them. So one thing about muscles that I want to at least cover is that they're contractile, which is unique, so they're able to shorten once they're stimulated. Then after they contract, they are able to return back to their original resting length. So they'll shorten and then they'll go back to their original length. Again, we're going to focus on skeletal muscle, so remembering from chapter 5, which is our tissues chapter, our skeletal muscle is striated, so having the alternating light and dark bands, that's what causes these striations. Later we'll talk about the actual proteins that cause this overlap and therefore this difference of color. Skeletal muscle is also voluntary, so it's the only one we're able to control with our own brain. And the cells that make up skeletal muscle is a myofiber. So let's look at this actual muscle fiber, which is just basically a muscle cell. So on the outside of our muscle fiber, we have the plasma membrane, and it has its own special terminology. So with your muscle cells, they do have some of their organelles and parts have their own special vocabulary. So up top we have the sarcolemma, so that's the plasma membrane of our muscle fiber. Inside of our sarcolemma is the sarcoplasm, so just like the cytoplasm in a normal cell. So this is going to have your fluid and all of your organelles in there. So inside the sarcoplasm we have our myofibrils. These are our contracting proteins. And we have our thick filaments, which is one type of myofibril. It's made of the myosin protein. The thin filaments is made up of the actin protein. Also located in our muscle fiber cell is glycogen. So remembering that glycogen is the storage unit for glucose. So that allows for our muscles to always have a source of organic energy for making ATP. The other thing that they'll need in order to be efficient at making ATP is myoglobin. So this is the oxygen storage. So it's very similar to what you've probably already heard of, a molecule called hemoglobin in our red blood cells. So its job is just for oxygen storage. So one unique structure or feature of skeletal muscle cells that very few other cells have is that it has multiple nuclei. So when the precursor cells fuse together, the nuclei don't fuse, and so you end up with having multiple nuclei. So those cells would be our myoblasts. Also in our muscle tissue, we do have satellite cells that will eventually become myoblasts, so you can make more skeletal muscle cells. So let's say you have some kind of damage to your muscle tissue. It's these cells that are going to replace your skeletal muscle cells. Mitochondria is still called mitochondria, but we need big mitochondria to make plenty of ATP for contraction. So with our organelles of the muscle fiber, instead of having endoplasmic reticulum, we have sarcoplasmic reticulum. So this is a type of smooth endoplasmic reticulum that wraps around our myofibrils, creating the structure of our sarcoplasmic reticulum. So our little blue cages here, and I have a better cartoon later, but these little blue cages, they wrap around our myofibrils. And at the end, we have this structure called the terminal cisterns. So the job of the terminal cisterns is to collect and store calcium, and it is released in order to activate our contraction. Another feature near our terminal cisterns is our T-tubules. So this is where the sarcolemma, so our plasma membrane of our muscle cell, has this infolding of our plasma membrane to create a hole for allowing our action potentials to travel into our cell. So these three structures, our terminal cistern, T-tubule, and other terminal cistern on the other side of our T-tubule, create what's called the triad. So the function of this is for when this active potential goes into our T-tubule, calcium is released from the terminal cisterns of the sarcoplasmic reticulum. That calcium then goes down its concentration gradient in order to activate the contraction. So we'll see later when we talk about our regulatory proteins, the binding of calcium is what's needed. All right, time for exam labeling. So with our exam, for this picture at least, you'll have other stuff, other things to label. We have the mitochondria, the nucleus, sarcolemma, sarcoplasmic reticulum, and Z disc. So our mitochondria in this picture is folded on the inside here, stuck in between those myofibrils. Nucleus, so again, they're on the outer edge near our sarcolemma. Next one, sarcolemma, so all of this yellow area, it is labeled here. So that's our plasma membrane, our sarcoplasmic reticulum, labeled here. So our little blue cages going around our myofibrils. And then a Z disc. So later we'll talk about what's the importance of the Z disc, but our little zigzaggy line right here. Okay, so let's focus on our myofilaments inside the muscle fiber. So we have two different types of myofilaments. We have thick filaments, which is made up of myosin. So that is the protein that creates our thick filaments. And the shape of our myosin molecules is kind of like a golf club. So the handle portion that you hold is the tail region. The head portion is kind of like the end club portion that you would smack your golf ball with. So in our thick filament, which if you look at the bottom of the slide, these purple golf club shaped looking proteins are arranged in a hollow tube, and the heads are sticking off of that hollow tube and away from the center of our thick filaments. So having those heads sticking out is actually important to the function of contraction. Okay, the other type of myofilament is a thin filament and is made up of the actin protein. So each of these little red balls is a actin protein. So on our actin protein, there is a binding site where the head of a myosin molecule can bind to it, and that is part of what is needed in order to pull on these thin filaments and cause the shortening of our muscle cells, so causing the contraction. Associated with these two thin filaments are two different types of regulatory proteins. So the first one is tropomyosin. So tropomyosin is this big, long, white structure here, protein. It blocks your active sites on your actin subunits. So it blocks the ability for that myosin head to be able to bind to actin. The other regulatory protein is troponin. Troponin is where calcium binds to. So earlier when I talked about the sarcoplasmic reticulum, when calcium is released from there, it goes to bind to our troponin molecule, and that helps with part of our muscle contraction regulation. Here's a slide that talks about the actions of those two regulatory proteins. So again, calcium binds to troponin in skeletal muscle. It binds to a different one in smooth muscle. We'll talk about that later. So when calcium binds to troponin, troponin is going to has a conformational change, so it's going to change shape, move the tropomyosin off those active sites, and therefore allowing for myosin to bind to the actin filaments. Going back to our thick and thin bands. So with our thick and thin bands, so I'm going to point it out in the picture at the bottom. So our big, thick purple ones, those are our thick bands. These orange looking ones are the thin bands. So with our striations of our skeletal muscle, they're caused by it's caused by the overlap between these thick and thin bands. So this section where the thick band is, and it overlaps our thick filaments that overlap the thin filaments. Where that happens, it's our dark band. It's also called your A band. So with this dark band, that's just part of what's causing your striation. So it's the darker line in the striation. The lighter line in the striation is where you have your Z disc as well as just your thin filaments. I don't care about you knowing about the elastic one, which is the green ones. It's not on the test. But I do want you to know we're in our I band, which is our light colored band. We have our Z disc, and the Z disc is the anchoring point for our thin filaments. Okay, so with our Z discs, they mark off our functional contractile unit of muscle fiber called the sarcomere. So from Z disc to Z disc, that is our sarcomere. When your thick filaments, so our purple here, when they pull on the thin filaments, they're going to pull the two Z discs closer towards each other, and that causes the shortening of the sarcomere, and that motion is transferred to your rest of your muscle cells. So you'll have multiple sarcomeres shortening and contracting together to actually get your muscle contraction. So your filaments don't change length during your contraction or your shortening. It's only the amount of overlap between your thick and thin filaments. Moving on to actually stimulating our muscle. So we're going to focus on motor units and therefore muscle contraction, looking at the neuromuscular junction between our neuron, nerve cell, and our muscle cell, and then look at how our stimulation works, so our membrane potential as well as the action potential that is triggered. So starting with the motor unit. So from our brain, we have somatic motor neurons. We have a upper motor neuron that's going to synapse or make a communication connection between a lower motor neuron. So the ones that are going to target a muscle fiber, the cell bodies are located in either the brainstem region or the spinal cord. So from our brainstem or spinal cord, the signal is sent through the axon, this long extension off of the cell, and towards our actual muscle fiber, so towards our actual cell. In some motor units, you'll have one neuron to very few muscle fibers, and you'll have some where it's one neuron to a whole lot more muscle fibers. But your muscle fibers are only going to be targeted by one neuron. So you won't have multiple commands from multiple different muscle fibers. It'll just be from one. So you get that command from our brain. So again, our voluntary control. It gets sent to our muscle fiber cell to stimulate it, to cause it to contract. So for our motor unit, it's one nerve fiber and all of the muscle fibers that are innervated by it. So when our action potential sends that command, all of the muscle cells or muscle fibers are going to contract in unison. So it's able to provide contraction over a wide area. For even stronger contraction or more effective contraction, you sometimes will actually have to have several motor units contracting at once, so sending it to multiple locations. Last piece for this video is the neuromuscular junction. So this is where our nerve cell, so our neuron, is going to signal a command to our muscle cell. Okay, so more vocabulary. So the connection site for communication between your nerve fiber or neuron to whatever target cell it has is called the synapse. So for our connection between a neuron and a muscle cell, it's called your neuromuscular junction. So just a specific name for that type of synapse. Okay. Just looking. Okay, cool. So when your nerve fiber stimulates your muscle cell, it can actually stimulate it at several points. So you have our axon terminal, so our end piece of our neuron. And inside that axon terminal, we have synaptic vesicles that have the neurotransmitter acetylcholine. So this type of chemical is going to stimulate and bind to receptors on our sarcolemma of our muscle cell in order to trigger what's called an action potential. So this is going to be the electrical charge that's going to stimulate the muscle to do the actual contraction. Also in this area, we have the synaptic cleft. So this is the gap space between our two cells. And then helping to keep everything together is a Schwann cell. So when we have a nerve impulse, a command from our neuron, it causes exocytosis in those in that axon terminal to release the acetylcholine in that synaptic cleft, so that gap space. This causes the release of that acetylcholine to bind to millions of acetylcholine receptors on our actual muscle cells. So helping to put all those receptors in the same spots. We have junctional folds. So just like we had microvilli in a normal cell, we have junctional folds making more surface area. Also in this area is acetylcholine esterase, so ACE, A-S-E for enzyme. That is going to break down acetylcholine so you don't continue to trigger the muscle when you don't want to. So here is picture form of our neuromuscular junction. We have the swelling of our end piece of our neuron, our axon terminal. It's filled with acetylcholine. Has those synaptic vesicles ready to go through exocytosis. So here is our action potential. It causes the release of the acetylcholine to bind to those receptors. When acetylcholine binds to the receptors on the muscle cell, it causes another action potential on the surface of your muscle cell. And we're going to start here for the next video.